All right.
All right.
So welcome to the Open Source Antibiotics meeting on Friday, January 22nd. We're getting together to talk about the mechanism of action work, where we just received data from Lee Graves' lab, who was on the call. And the aim of today's meeting is to talk through that and think about targets for potential targets for the molecules that we're interested in. Just as a very quick mention of stuff that followed up from last time, just very briefly, because I ought to mention it. So we just, in the last couple of weeks, we've spoken about a bunch of different molecules that may be of interest for the next round of synthesis and evaluation. We discussed those, with words, and I'd written some notes, and Dana's very kindly put those into a diagram so that we can just refer back to this as these being the current synthetic targets. And I think it encompasses everything. We left one of yours, Anthony, but I've stuck it up there now, the thiol. And there are a bunch of that, as well as something about the one-pot method and a shopping list that Dana's putting together to get molecules in the lab ready for when she arrives back in London. So hopefully that's clear. And I don't really want to discuss that anymore because we've discussed it quite a bit, and there's some really good targets there that I think are very achievable. So I think that in terms of the chemistry planning, we're in good shape. And really the highest priority item is the MOA stuff. So we obviously received the data from Lee, and Dana's put up this explanation of what that is, as well as some the sort of highlight targets from the sheet there, and then Giada put up some links for the relevant proteins. But that's kind of as far as we've gotten here. And what I was hoping, Lee, is you could enlighten us on both things, the meaning of the data and what we should be taking away, and then what we ought to be doing next in terms of follow-up experiments.
Mm-hmm.
Actually,
Yeah.
Matt, would it be okay to maybe just give us a brief run through of how the experiments are done?
Oh yeah, that would be fantastic. If you wouldn't mind, Lee.
Oh yeah, I'm going to try to remember exactly what was done because it's been a little while now. And my technician that actually did the work is in another meeting right now, so he couldn't join us today. But the idea was the inhibitor bead cocktail we have is a broadly effective agent at capturing kinases and MAS. So basically, we've used it as a way to study the kinome from a variety of different systems. This is the first time we've ever applied it to a microbial system. But the idea is that these compounds bind promiscuously to the ATP binding site. And so we can capture a large number, usually 300 or so kinases. And then using quantitative mass spectrometry, we can look at profiles of kinome differences. What we found to be quite effective is looking at competition assays, using this platform as a competition assay to look for drugs that compete off kinases in a dose-dependent manner. So basically the idea was, let's take some staph A cells and then incubate them with your compounds at different concentrations. And if I remember correctly, that's what we did. We did both the active and inactive compound that you gave us, and a lab next door prepared the lysates. So we incubated the lysates with that, and then looked for what things were competed off in a dose-dependent manner. So the things we tend to look for is, how significant were the peptides? Did we get 2 or more? Were the fold changes reasonable? Did it show dose dependency? And then not dose dependency with an inactive analog. So I really haven't gone through this data because I don't know these proteins very well. But I was hoping that when you guys looked at them, you'd find things that were of interest to you. And I will add one more thing. We find that these beads oftentimes bind other proteins that are not kinases. If the ATP binding site is occupied by the inhibitor, we can still see competition-dependent binding, and that's sometimes quite important in terms of the targets we're finding. For instance, this enzyme called FETCH, which is involved in heme biosynthesis, we found that, and other people have published this as well, that oftentimes kinase inhibitors capture FETCH and other metabolic enzymes, which you wouldn't necessarily look at. But I asked my technician to give you all the data, not to filter it just for kinases, just give you everything. So I'm anxious to see what you guys actually have come up with from that list.
So on your beads, do you only have reversible inhibitors? They're not covalent ones or anything like this then? Yeah.
Okay. Yeah, they're all reversible.
And these would also presumably all be inhibitors of mammalian enzymes.
Yeah. But they're very promiscuous. They're almost sauersporin-like, so that most proteins.
Okay. Yeah. So are they structures in the public domain anywhere?
Yes, and we can give you all those structures if you would like to look at them.
It would be very interesting just to see if any of them look anything like the ligands as well.
Yeah, no, that's a good point.
Yeah.
I can have that sent to you guys.
That's brilliant.
You know, the key for us was just being able to put an amine group on the end so we could attach it to a bead, or in some cases carboxyl. But it really didn't affect the properties of kinase binding.
Yeah.
So I guess we had a question last time, and please excuse any ridiculously naive questions. We had a question last time of the fact that if this is the first time you've applied it to a microbial system, we were wondering about the target list that you came up with. And you know, these are inhibitors of mammalian enzymes. The list you sent us of codes, these are for, we were confused, I guess, about where it crosses over from mammalian proteins into microbial proteins. I guess that's what we were wondering. So how the targets you've listed there, those are being fished out as mammalian proteins?
No, they were, what we sent you was just basically whatever the mass spec detected as a significant change in amount bound to the bead. So it was all bacterial proteins. Shouldn't be any other, maybe there was occasional contamination of a keratin, which happens sometimes, but it should have been all bacterial proteins in there. And, you know, we didn't do any kind of other analysis besides give you the data, basically.
Okay, great. I mean,
so were there any proteins that you couldn't identify that were upregulated?
Oh, well, you know, we didn't do a total proteomics analysis, which would have been an unbiased approach. We could still do something like that.
Because I presume bacterial proteins are not as well characterized.
Right. Yeah, no, that's a very good question. You know, like I said, this is sort of our first foray into this organism. So they're probably, you know, we rely on the libraries, the databases that are available, and bounce our data off that. I think they're pretty good for staph A, and that's, you know,
okay.
Shouldn't be a lot of proteins that weren't identified, but I can ask them that too.
Yeah, I think this is really fascinating. It's quite a steep learning curve, though.
Well, and, you know, like I said, it wouldn't probably be a bad idea for us to do some just unbiased proteomics where we treat the cells, living cells, with the compounds for various time points and concentrations, and just ask, how does the proteome respond to this? And we've done that in other systems and learned a lot about potential targets, you know, potential effects on metabolism and so on, without invoking the beads at all. Just ask, you know, how does the proteome respond to this? And that can be quite useful too.
Yeah, I had a question to do with that. I mean, my first thought was that we should look at the genes which have been, sorry, the protein levels which have been affected, and ask if any of them cluster and are, you know, related to a particular pathway in the cell. But of course, that wouldn't apply here because it's a lysate. So it wouldn't be that you, right. Whereas if you do the experiment you just mentioned, then you might pick that up.
Well, and we picked the safe route.
I'm sorry, would it be possible for us to make a compound that's functionalized that could be tethered to a bead? So we have our drug tethered to a bead, we run lysate through it, and we pull whatever we can get out of there and look for that?
Yeah, it's a very common experiment, and the difficulty is finding a place where you can do that. And so there's quite a bit of work involved in figuring out the best attachment point. So
Yeah,
this, I guess, gets over that issue that you don't really have to worry about that so much.
Mm-hmm. But the assumption is that the protein of interest that you want to follow is binding to our beads, and there's a possibility that it's not.
So just, yeah, on the list here, I mean, I guess I wondered, from the ones that have been highlighted here, which are the ones that we've picked out, I wondered if there were any that are typical false positives, you know, that tend to get picked out all the time. I know I'm putting you on the spot here. Sorry.
Yeah, no. I'm looking at it. I don't see any that I would say, yes, those look like odd or ones we've seen in other examples. No, they're quite unique, actually.
Okay.
You know,
and what was the? Sorry, Dana, you picked the ones out, right, that we were going to look at here, and I'm sorry, I forget what the oh, you've got the criteria here for why you selected those, right?
Yes.
Yeah, so these are the most affected.
Right. So the 0.5 or negative 0.5 or plus 0.5, that was from sort of the PowerPoint that you guys supplied us with, Lee, to say that this is significant. So I looked at that criteria. I just said at the highest concentration that those criteria were met, and then I looked for dose dependency. So that's what I got, and I've gone through a little bit of the links that Giada found. So some of these do have ATP binding sites, so I think that would kind of make sense. So I guess my next question is kind of, is it known that any of these are essential for bacterial cell survival? And that I'm not sure about.
I'm pretty sure a couple of them are like extremely important. There are some pyrimidine synthase in there. As you know, the sulfonamides target DNA synthesis and cause cell shutdown through there. As you can see, there's a couple pyridine and pyrimidine synthesis proteins, such as the PYR, not PYR. It should be up a bit further. There is a phosphoformoglycinamide. Pure Q is a DNA synthesis one. Similar to what sulfonamides target, except they target folate instead.
Is lipoyl synthase also some people have targeted for antibacterial? I don't know why I'm thinking that, but one of my colleagues works on Neisseria gonorrhea.
I think lipoyl synthase is used as a post-translational protein modifier. I'm not sure.
Okay, I mean, so that's something that I think we probably need to do more literature searching on, just so that, you know, we know that there's evidence in the literature that these targets are, you know, could potentially affect the bacterial cell survival. But Lee, I have another question about the experiment. So some of these ones that I picked out show a positive fold change. Am I still here?
Right. Yeah, that's a curiosity. And we see that with certain metabolic enzymes. And I never had a good explanation for that. Now, there's two possibilities. One is that through the competition, we're now pushing off some protein that allows binding sites for one of these other enzymes to preferentially bind, right? So that if you imagine that some, because of avidity, there's some proteins bind to the beads, and as we compete them off, now we provide binding sites for other proteins to bind. That's one possibility. The other is that we're affecting an allosteric site, that we're binding to an allosteric site and then increasing its affinity for the beads. And we've seen this with a lot of metabolic enzymes, which makes me think it might be the latter, that you're affecting some kind of allosteric site such that it now prefers to bind to the bead even tighter. But I don't have a good explanation. In essence, it should be simply a competition experiment, and we should see proteins decreasing. And, you know, it's a very, it's a difficult question to answer.
Okay, so I guess actually my first thought was neither of those. So my first thought was that maybe the inhibitor was targeting like some upstream target that would then increase the biosynthesis. Does that make sense, or would that not happen because we're using a cell lysate?
Yeah, Dana, we're using a cell lysate and we're using a short exposure, and I think it was no more than an hour. So that's very possible if you're treating live cells. But we chose not to do that because we weren't sure if we were certain that the compounds were getting at the concentrations that we wanted to see competition. So we chose a safe route, which was to make the lysate and do the experiment that way. I actually prefer doing it with live cells because, you know, you're looking at competition with relevant ATP concentrations. You haven't disrupted some of the scaffolds and complexes that are naturally found in a cell. That can be the next line of experimentation, possibly, to see if we can repeat this with live cells. Now, Dana, when you looked at the data, did you say, okay, the positive control compound gave assist, the negative control did not show a change, and use that as part of the criteria for analyzing these proteins?
Kind of. So yes, I looked at the ones that showed significant fold change at the highest concentration. Then, oh, right. So then I filtered out anything that didn't show a dose-dependent change with the active compound.
Okay.
And then from that list, I filtered out anything that did show dose-dependent changes with the inactive.
Right. Okay.
Does that make sense?
Yes.
Okay.
Excellent. Yeah.
But also, so positive and negative regulation, was it, or did you just shortlist the negatives?
No, I just said dose dependent. So I didn't distinguish which direction it went. So I was kind of tabulating them all together. It looks like a lot of them are actually positive fold changes rather than negative.
Oh, yeah.
So that's kind of interesting.
Yeah.
Any that you can say for sure were negative then, because I think those are the ones we might want to focus on first.
Okay.
That's interesting.
Yeah, so the MQ01, yeah, there are a couple. PURD, some of them, yes.
Okay, so you'd recommend, if we were going to try and investigate this directly, you'd think that we should maybe look at the ones that are negatively impacted, negatively regulated.
Yeah, I would start with that.
Okay, if there's a shorter list, that also helps.
Yeah. And if there's antibodies available to any of them, we can take our remaining samples and just do a Western blot and see if we see competition using Western blot analysis as well.
Right.
That's a quick follow-up.
Do we have a list of the negatively hit proteins, or?
I can put it together. I have it just in an Excel sheet on my laptop.
Okay, because I'll do what Giada did in my 9 Uniprot, I guess, for them. It's going to be a shorter list, so shouldn't be too hard.
And I mean, obviously, if any of these are already known as targets for, as Lee was saying, for antimicrobial drug discovery, then that will be a place to home in case someone already is looking at some of these enzymes.
Mm-hmm.
Well, we can cross-reference the Uniprot codes against Kemble.
Oh yeah, great, okay. Yeah. Okay. And zoom in on things that are known and then things that are unknown, and then we're in better shape. The one remaining question I had about the data also is, so the level of activity that was seen. It's not as if we're dealing with a relatively small amount of influence. Are we dealing with experimental values that are typical, or is it near noise? Are we talking about significant impacts on some of these relative to a typical experiment, I suppose?
I'd have to bring up the raw data. I thought there were some though that fit that criteria.
Again, I'm just unfamiliar with the experiments.
That was PCE 780.
Sorry, you want to look at these?
Yeah, there were some that had fold changes. I mean, higher than 1, and even some of them that went up to 2 that were still sort of dose dependent, but nothing in not as many as in the negative direction.
Oh, yeah.
There's a couple that get to like -1.4, -1.1.
Again, I'm just interested in if that's typical,
I think it is. Yeah. Sometimes we see more significant changes. I'm just digging up. I found this file here.
Okay.
I think one of the things is very valuable is to have a positive and negative control like you guys have. This is not the file I want.
Yeah, we're swimming in negative control.
Yeah.
This molecule has been extremely finicky.
And I forgot what 26 was 822. I've got the numbers. What is the active and what's the inactive here?
The active is 822.
822, and that's also known as 28. Okay.
Oh yeah, that's the original numbering. Yeah, right.
Yeah, there were some significant fold changes with some of them, and it may be too, in some cases, we won't see such nice dose dependency. It'll be like a threshold. We hit a threshold and only at the highest concentration we see something. I probably should just take a little bit more time and go through the data too and just bounce back with Dana if I see anything else that might be important in there.
Yeah, I mean, if you wouldn't mind, that would be really helpful because, of course, we've got to think about what to do now.
Yeah.
In terms of publishing, in terms of getting the story straight, we want to try and validate something.
Yeah, and let me talk to my tech too. He had told me that, and I don't remember if it was that he wasn't happy with the amount of protein, that we didn't have quite as much protein as we would have liked to have had for the experiment. So it might be worth our while to just go ahead and repeat it with a larger amount of protein and try and get more proteins into the mix. That often makes a difference in terms of the results.
Okay.
But let me chat with him today about that.
Okay. All right, great. All right, fantastic. And then, let us know what you think and also your interpretation of the data. That'd be very helpful.
I mean, we always have to do these things over, like any experiment, just so we're certain with the results, and then to start digging in, knocking things out, validating with Westerns and so on, which,
Great. Level. Okay. All right. Does anybody else have a question for Lee while we have him on the call? Otherwise, I think that's everything we wanted to talk about today, right? So we can do our analysis of the data, and we'll stay in touch with Lee about sort of follow-ups.
All right.
So we'll approach both directions. I
will send the structures of the compounds to the group.
That'd be wonderful.
So we can take a look at those.
Yeah, great. Okay. Was there anything else anyone wanted to raise while we're on the call? Because this is the most important thing.
No, I think you've given us a lot of food for thought.
Yeah, it's great.
Okay.
It's great. Like I said, Lee, the 3 parts of this project are getting the potency better, and that was all the chemical structures you saw at the beginning is our best guess of what to change next. And then trying to get rid of some of the metabolic liabilities, which is the stuff we're doing with Monash. We've done a few experiments on compounds with them, and we're trying to get a sort of next best compound so that we can send them those towards the end of the project.
Yeah, it would be lovely if we could, if it was some protein that structure was already known.
Yeah.
So we could just like see it folded at home, or someone can just do like docking.
Yeah, that would be great. So let's see. But at the same time, novelty's good.
Yeah.
Well, and, I'm going to say that, the biggest danger with our method is that the protein that you're looking for doesn't bind to these beads.
Yeah.
And so we don't ever see it. And so we may want to step back and say, well, how can we do an unbiased proteomics analysis to make sure we're not missing something very important?
Yeah, sure. Yeah.
And that's, I don't want to let you guys down.
Yeah.
All right, that's great. Thanks very much, Lee, and thanks everyone for coming along. And we'll pick this up again next week.
Right.
All right.
Sounds good.
Nice to meet you all.
Bye, everybody.
And you. Bye.
Bye.